There we go.
All right, so thank you. Hi, Doug. How are you? So, welcome. I'm sorry I can't be there in person. So, it's my pleasure to briefly introduce Doug Walker, who's very well known to the Emory community. I mean, Doug got his PhD from Tufts, and he's just telling me that most of his dissertation was done here at Emory, working with Dean Jones and largely in metabolomics. But Doug then moved on to Mount Sinai, from where Emory recruited him back to out here, and it's a Department of Environmental Health where Doug has his primary home. He collaborates also with the Cancer Center and also with the Emory Global Diabetes Research Center. We were very excited to do a little modest support to help Doug join us. So we think there's tremendous potential from the work that Doug is doing, particularly in the area of measurement of exposome and some of the work we do as they relate to diabetes, both at the neighborhood level and at the individual level. So I'm hoping this seminar today is going to provoke a lot of interesting ideas and potentially lead to some very substantial collaboration with Doug, both immediately and in the long run. So with that, Doug, I'm going to take it away.
But as Venkat said, I'm delighted to be back here at Emory. I spent about 5 years here at Emory working with Dr. Jones in his laboratory in the pulmonary division, where I really had the opportunity to be introduced to metabolomics and the exposome and really start developing my research interest in environmental health. I'm actually an environmental engineer by training. When I started my PhD, I was originally hoping to solve all of the groundwater problems that we had in the world, but I quickly came to the realization that they're unsolvable and was very happy to have the opportunity to move into more health-focused studies, and very fortunate to have a great mentoring network here at Emory, including Dr. Jones and Dr. Gary Miller when he was here before going to Columbia. All right. So just a little bit about my background before returning to Emory. So after I left the clinical biomarkers laboratory in 2018, I joined the Icahn School of Mount Sinai as a faculty member in the Department of Environmental Medicine and Public Health. And there, my research goal was really to take these exposomic approaches that I've been using in Dr. Jones's laboratory and try and optimize them for large-scale screening of the human exposome and of within human populations. And so a lot of our focus was really on operationalizing these approaches for measuring the exposome, as well as developing approaches to measure exposures of emerging concern. And these include things like microplastics and PFAS and other perfluorinated compounds. So really, we were trying to use the instrumentation that we leverage for exposome research to not only understand exposures in human populations, but also advance the methods to measure new exposures within those as well. So since returning to Emory, I've been working on establishing the comprehensive laboratory for untargeted exposome science. And our goal is really to integrate advanced analytical equipment for measuring the exposome into an approach that allows us to both characterize exposures that could be leading to increased risk for metabolic disease. And so we focus a lot on cancer and diabetes and obesity-related diseases. We're also, again, using these approaches to develop new biomarkers for understanding exposures of emerging concern. A lot of our emphasis is on microplastics right now because there's a lot of interest in understanding these exposures, but currently there are not any good methods for measuring them in human tissues. And also, we're trying to develop approaches that allow us to prioritize exposures that could be having an adverse outcome. So rather than looking at how an exposure is linked to a disease outcome, we're trying to develop strategies that allow us to identify exposures in human populations that may be causing a change that leads to increased risk of developing a disease. And so we do this through a number of different approaches. Some of these are trying to integrate multi-omics into our studies so we can see what are the series of biological changes that occur in response to an exposure. as well as developing approaches that allow us to use bioactivity assays to understand which exposures may influence a change that could lead to risks for things like cancer or metabolic related outcomes. So today I'm going to focus on really three main areas during my presentation. So first, I'm going to spend some time defining measurement of the exposome from a practical perspective. So many of us are familiar with this concept of the exposome, but the exposome as a whole is very complex and may not be measurable. So what we're trying to do is really understand what are the key measures of the exposome that we can provide and how these can be used to better understand the relationship between environment and our health. Next, I'll talk about the high-resolution mass spectrometry frameworks we're developing for large-scale exposome epidemiology that can be applied in population studies as well as mechanistic studies of diseases. And finally, I'll spend some time discussing looking to the future and the Human Exposome Atlas project. So how can we systematically characterize the exposome and relate that to multiple diseases and comorbidities? So following completion of the Human Genome Project, there was this anticipation that genetic testing would really revolutionize our ability to understand what drives disease risk. We thought that we could potentially go to the doctors, we could have a full genotype worked up, and the doctor would be able to tell us that we have these mutations or these variants, and these give us X percentage increased risk of developing a disease. But what we found over the past 20 years is that the reality is much, much more complex than that. We know now that genetics is a contributor to our health, but it is not the only contributor. And there's a number of different factors that drive our risk for developing diseases. And this is very, very evident from this study that came out from Steve Rappaport about 8 years ago now, showing the population attributable fraction for common disease phenotypes from monozygotic twin studies. And so the population attributable fraction is essentially the percent reduction in a disease or percent reduction in a prevalence of a disease if we account for some risk factor. And since this is a study of monozygotic twins, in this analysis, they're essentially evaluating gene or gene by environment risk factors. And so the main take home from this, when you look at the plot, is to see that most of these diseases, which again are very common chronic or age-related diseases, only have a population attributable fraction from these monozygotic twin studies of about 30% at the most. Most of them are less than 20 or even less than 10%. And so what this is really telling us is that when we consider these gene or gene by environment risk factors, it's really only accounting for less than 30% of what's driving disease risk. And in fact, our current estimates that suggest that really only 5 to 15% of diseases can be attributed to your heritability or your genetics alone. The other 85 to 95% is really derived from environment, lifestyle factors, and its interaction with the genome. So if we take a genetic only or a heritability only based perspective of human health and disease, we're really applying a very unbalanced view of what's driving disease risk in humans. And so this, let's hide that. And so this need to better account for environment led Christopher Wild, who was the director of the International Agency for Research on Cancer, to initially define the concept of the exposome, which he envisioned as a complement to the genome. And when he first thought about this and conceptualized it, he essentially defined it as life course environmental exposures, including lifestyle factors from the prenatal period onwards. And so to use this definition in the purest form, essentially he was saying that we need to account for all non-genetic disease risk factors or health factors that could be driving outcomes in human populations, and that are not genetic. And we need to characterize them in a way that's comparable to the systematic measures we use for measuring genetics. Unfortunately, this is a very challenging idea to put into practice. Unlike the genome, which is relatively static throughout your life, your exposome changes daily. My exposome in the laboratory might be different than what it is here in this room, and it may be different at my house. And so unlike the genome, our exposome is, inherently variable and changes at different timescales. So this makes it very challenging to essentially measure the exposome using a framework like what we would have what we have for genetics. And so this need to, better understand or redefine the exposome in terms of pragmatic or practical measurements led Dr. Jones and Dr. Miller to really redefine the exposome in 2014 as the cumulative measure of environmental influences and corresponding biological responses throughout the lifespan. And while this definition may sound relatively similar to what Christopher Wilde originally proposed, there are some very, very key and important differences. The first is this focus on cumulative measures of environmental influences. So rather than defining the exposome as something that has to be measured at all time points, this definition really emphasizes what are the most important measures of environment that allows us to understand somebody's individual exposure history. And I like to think of this as kind of an analogy to standing on top of a mountain and trying to determine where the other mountains are around you when there are clouds kind of covering the base of the mountains. To know where those peaks are relative to you, do you have to know exactly where each mountain begins and the other one ends? Or do we just need to know where those peaks are, and to just understand their position, we just need to know where they are above the clouds? And that's very similar to the philosophy that we take with trying to measure the exposome. Rather than getting caught up on this definition of measuring everything, what are the key measurements that we can develop to understand somebody's complex exposures, either at a certain time period or potentially throughout their lifespan? But the challenge is really, where the rubber meets the road, to say. So, we've seen tremendous advances in technologies that allow us to very comprehensively characterize biology. It's now possible to have your whole genome sequenced at the Emory Genomics Core for about $700. You can get genotyping done for under $100. 23andMe is an example of genotyping that's being used where common mutations and SNPs are being measured. We have next generation sequencing methods to very comprehensively measure gene expression or gene methylation, as well as a lot of advances in proteomics platforms that allow us to screen for thousands of proteins in a biological sample. However, our ability to characterize environment is still using technologies that have been applied for the past 30, 40 years. Most of our current technologies only allow us to measure about 10 to 100 exposures. And so obviously there's a discrepancy between our ability to characterize environment relative to its importance to our health. And so what drives the research in my lab and the research that my excellent team members who are sitting in the back are doing is really trying to address this critical need to develop analytical frameworks for universal screening of the exposome. So how do we develop approaches that go from measuring 10 exposures or 100 exposures in a population to developing the analytical strategies and approaches to measure tens of thousands of exposures, which we know is something that is experienced by all of us, throughout our lifespan. And so the way we've approached this is through really focusing on the internal exposome, because we know that for any exposure we come in contact with, it has to be internalized for it to really have an effect on us. And so we've approached this through developing strategies that allow us to very comprehensively measure the human metabolome. And at its simplest form, the human metabolome can be defined as the complete collection of small molecule metabolites found in the human body. These include small molecules like peptides, lipids, amino acids, nucleic acids, and all the other endogenous metabolites that are produced by our body so that we can maintain proper homeostasis. But at the same time, what's unique about the metabolome and different from the other omic measurements that we can perform is that the metabolome also includes things like food additives, drugs, toxins, pollutants, and just about any other chemical that humans ingest, metabolize, catabolize, or come into contact with. And so that's a very, very critical aspect of the metabolome that makes it feasible for us to use it for these exposome approaches. Not only can we measure changes in endogenous metabolites, which tells us about the effect an exposure may be having, but we can actually measure biomarkers of the exposures themselves when we're characterizing the metabolome. And so the metabolome really provides a critical link between exposure, biological response, and disease outcomes, and really fulfills the needs of the measurement of the exposome based upon that definition proposed by Dr. Miller and Jones. And so within the metabolome, we can not only characterize internal dose biomarkers, which tell us some of the exposures that an individual could have come in contact with, but we can also see how those exposures are associated with changes in endogenous metabolic pathways. So that tells us about the effect that they may be having. And at the same time, we know or can provide hypotheses about how some of these biological changes predispose you for developing diseases. And so we can then look at how those exposures are linked to altered metabolic pathways that may be consistent with increased risk factors for diseases. So again, it really provides a singular measure that can potentially cover that continuum from the exposure to the disease phenotype. And so our current estimates suggest that, we know from our estimates and from years and years of study that the metabolome itself is very, very complex. So when we talk about characterizing the metabolome, our current estimates suggest that over 300,000 chemicals or signals could potentially arise from external sources. So these include things like food constituents, drugs and drug metabolites, as well as commercial products, environmental chemicals, and their metabolites. And at the same time, we can measure a very large range of endogenous metabolites or compounds that are produced within our bodies. Some of our estimates suggest that this could contribute up to 600,000 to 700,000 different chemicals to our metabolome. And so when we think about measuring this, we recognize that we can't develop an approach where we will know exactly what we're measuring ahead of time. So if I try to set up a targeted method that says measure all million of these compounds, it would never be possible to do that. But we still want to be able to characterize what's in our samples as much as possible and really use data-driven approaches to prioritize what are the key measures that we should be detecting in our studies. And so really to understand and embrace this complexity, we have to develop analytical strategies and study designs that incorporate non-targeted measures of exposure. And so let's talk about what that means from a practical perspective. So, of course, it's all well and good to talk about doing this, and it's a very, inspirational vision to talk about measuring the exposome. But the reality is that this is very, very complex, and we need to develop and adapt the strategies that allow us to characterize these exposures. And so in terms of implementing the exposome, our main goals or priorities have really been to create what we like to think of as an open and fair exposome analytical framework. And we want this to be able to allow us to perform robust and large-scale profiling of the human exposome. We want to make it a reality to analyze tens of thousands of samples using these approaches we've developed. A key aspect of this is improving accessibility for environmental health and biomedical researchers new to exposomic research. So it's not good enough to develop data if only a few people know how to use it, or the learning curve is so steep that you have to become an expert in it just to analyze the data. We really want to make the large-scale exposome data we're generating usable and accessible to anybody who's interested in applying it into their research projects. We want to do this in a way that helps us establish a cumulative exposome disease atlas. So we don't want to be thinking just within one study. A lot of the previous metabolomic studies that have been done at other institutions, not necessarily here at Emory, but at many other universities, tends to think of each study as its own separate entity. So if I'm looking at a study of diabetes with 1,000 samples, I run those 1,000 samples, I analyze the data, write my paper, and upload the data to repositories, but they're still their own separate study. But there's value in looking at a diabetes study here in Atlanta versus a study in India versus a study in California. And so we like to think of each study that we're analyzing as part of a whole that will contribute to developing a large cumulative resource where we're now integrating across multiple studies, across multiple diseases and multiple populations. So we can really think of looking at similarities and differences across many different diseases and many different populations. And finally, I'm not going to talk much about this today, but another main goal is really to turn the paradigm around for some of our exposures and stop studying things from an outcome-centric perspective and start thinking more from an activity perspective. And what I mean by that is a lot of our studies, we will go out and talk with different cohorts all over the world. They'll say, okay, I have 1,000 samples from people who went on to develop diabetes. And these samples were collected before they were diagnosed, before they were showing the phenotype. And then I can match those to 1,000 controls. And so we have 2,000 samples, we analyze those, we see what's associated with diabetes and why could that be. But that's very expensive. It requires the right cohorts. It requires access to samples. And so there's a lot of challenges in finding appropriate cohorts for many of these questions for other diseases. And so the question is can we actually measure what's in humans and then try to relate those to disease outcomes by looking at how those exposures may change biological activity? But again, that's kind of a longer term goal and not something I'll talk about as much today. And so again, you can see that this challenge we face is really complex. And this is just an example of the complexity of our exposome. So in this study, the authors were hoping to try and do an accounting of all the different chemicals that are used just in plastic manufacturing. These can be used at any point in the process, from manufacturing through synthesizing the polymers. So it's from the whole continuum, from getting the raw materials through generating plastics. And what they were able to find, the individual categories aren't that important here, but they were able to identify over 10,000 unique chemicals that are used in plastic manufacturing alone. And if you consider the millions of tons of plastic materials that are being produced each year, even if some of these chemicals are only used in a very small percentage, there's a very, very, potential for a very large exposure to occur. And again, so this is just one aspect of commercial product where these chemicals are being used. And so, we face a tremendous challenge in trying to understand all the different exposures that could occur to humans. And so we've really adopted this untargeted strategy, which is so key to allow us to, systematically and robustly characterize samples for large numbers of exposures. And this differs conceptually from traditional approaches in that we're not a priori identifying what are the biomarkers we want to measure in our studies. And so in a typical targeted study, we tend to identify a hypothesis we want to study. So let's say we think exposure to a certain pesticide may increase our risk for developing diabetes. So if we were performing a targeted study, we would develop a method that allows us to measure that pesticide in human populations. We would identify our study population, analyze the samples, test our hypotheses that this pesticide is associated with the outcome, and either provide evidence that it supports it or provide evidence refuting it. But we're very much focusing on that exposure and measuring that exposure within the population. And using these approaches, these are more traditional strategies, and we can only analyze about 10 to 100 exposures using these approaches. With untargeted analysis, we're essentially switching this approach around. So rather than identifying the things that we want to measure in a population, we're developing robust analytical methods that can be applied widely to the samples we want to analyze in a robust and relatively routine way. And then we're applying it to measure as many exposures and as many signals as possible within a population. And then with these measures, we apply different bioinformatic and biostatistic techniques to essentially tell us what are the key exposures that could be driving the outcome. So we're essentially removing our biases and blinders from essentially a predefined concept or predefined hypotheses, and saying what are the key exposures that we should measure in this, or identifying what those key exposures are in the population. And these approaches, which we've really implemented for our exposome studies, allows us to analyze 1,000 to tens of thousands of compounds. And so how do we actually do this? And what does this look like in the lab? So our assays and analysis are primarily based upon or based around a technology called high resolution mass spectrometry. And so when we are developing our methods and analyzing our samples, we essentially have to take the sample and put it in a format that allows us to analyze it on our instruments. We have different basically pre-detection methods that allow us to simplify our samples to help us detect more things. And then the analytes in the samples that we're analyzing are essentially detected in the high resolution mass spectrometer. And I'm not going to go too much into detail about what that is, but the most important thing to know about the mass spectrometer is it provides us with two key capabilities. The first is that it detects a value, or it detects a relative intensity of many of the compounds that are in our samples. So while this won't directly give us a concentration, if we measure these compounds or ions in our samples, and we find that in sample A, it's twice as high as in sample B, we can assume that the concentration in sample A would essentially be twice as high. So it allows us to perform statistics and analyze our data to see what are relationships between the compounds we're detecting and an outcome. The second most important characteristic of these instruments is that it allows us to predict the identity of the things that we're measuring. So with the mass spectrometer, we essentially detect masses, and there's a lot of different ways that we can take that detected mass and translate that into a predicted chemical identity. And this is really important with our untargeted studies because, again, we don't necessarily know the things that we're detecting initially. And we want to be able to measure as many things as possible and try and determine what they are based upon our study outcomes. And then, of course, we also implement a number of different bioinformatic and biostatistic methods to identify what those top signals are. One of the very important things to keep in mind is that the mass spectrometer gives us numbers. And so we have to apply a number of different approaches we have to actually determine what those signals are. So anybody here that's worked with untargeted high resolution mass spectrometry data before may know some of the challenges that we face with actually identifying the signals that we detect. And that's something that many different groups are working on, including Dr. Jones's lab here at Emory. And so just as an example of how widely we've applied these approaches, and so if you can name a tissue, we've probably either already analyzed or thought about analyzing it using these untargeted approaches. The typical biofluids that we tend to use to characterize the exposome is plasma or urine, but we have studies on everything from brain tissue, different tissue biopsies, a number of different birth tissues like placenta and amniotic fluid, worked on stool, and so just many, many different tissues. And one of the advantages of these methods is that they can be relatively easily adapted and applied to different types of tissues. And this allows us to not only look across different organs or tissues to understand exposure profiles, but also potentially study the organ of interest that may be affected by an exposure or a toxicant. And so, I've mentioned previously how important it is to really be developing methods that allow us to measure as much of the complexity of the exposome as possible. And one of the challenges that we face with any analytical method is that the analytical method you're applying is inherently biased towards what it detects best. So a lot of our initial approaches have been developed using a technique called liquid chromatography with high resolution mass spectrometry. And these methods are very, very good at measuring polar compounds, but may not be very good at detecting semi-volatile or volatile compounds. And many of those semi-volatile compounds are exposures that we want to measure. And so to really improve the chemical diversity of what you can detect, it's very, very important to think of complementary or alternative analytical strategies. And that's essentially what we've done for these exposome methods, where we use different analytical approaches to really enhance or increase what we're able to detect. An example of some of the methods that we're currently using is through development of different gas chromatography methods that allow us to measure these volatile and semi-volatile metabolites through work done by Dr. Jones and Dr. Hu, who's also in the pulmonary division. They were essentially able to show in this recent publication that it's possible to develop a systematic, reproducible, and quantitative method for omic scale biomonitoring using GC HRMS, or GC high resolution mass spectrometry. And this was a very, very important advance because most of the exposome work to this date has really focused on liquid chromatography. And this allows us to expand to a whole other range of chemicals that can be measured using these platforms. And at the same time, combining that with liquid chromatography, high resolution mass spectrometry, we essentially have, with the LC approaches, we have a method that not only allows us to characterize metabolic products of many exposure biomarkers, but we can also characterize biological response signatures, which tells us a lot about how our body may be reacting to an exposure and how it could be predisposing us to a disease. And an example of how we're applying that is shown in this recent paper we published on benzene, where we were not only able to identify both known and novel benzene metabolites, but we were also able to identify alterations in pathways related to hematopoietic stem cells and hematopoiesis. And these are essentially the pathways that are altered in response to exposure to benzene and may describe some of the increased risk for leukemia that's been associated with benzene. But overall, it's really this combination of the two platforms that have allowed us to establish a sensitive, systematic, and robust measures necessary for performing what we like to think of as exposome-wide association studies. So we now have the functional measurements that make it possible to identify samples, identify studies, analyze as many exposures as we possibly can in those studies, and then perform association studies to say, okay, these are the top exposures or top signatures predicting future risk of this disease or development of this disease. And so, as I mentioned previously, a lot of our effort has been focused on taking these methods that were developed in Dr. Jones's lab and that we've worked with and really make them suitable for large-scale exposome epidemiology. And so our focus has really been on improving throughput, improving robustness, and making essentially scalable approaches that allow us to rapidly analyze samples and also rapidly scale up these capabilities. And so an example of how we've done this is through adopting fully automated open source sample preparation methods, where we can rapidly prepare about 96 samples each day. And this has both drastically reduce the time it takes to prepare samples and analyze them, as well as improving the robustness and CVs, because we don't have to worry about things like operator effects and other things that add some variation into your data. But the greatest part about this is we've essentially established a unified workflow where we can take a single sample, aliquot it to our different robots, and prepare it for all of the different assays that we perform to measure the exposome. And currently we're performing 5 different assays. 2 of those are liquid chromatography, or 4 are liquid chromatography based, and 1 is on the GC. And at the same time, a goal has really been to improve accessibility and traceability of our data. And so we've been spending a lot of time thinking about how we can best package our data for our collaborators. We work with many different collaborators in many different fields. And so we want to give them as much data as possible, but provide it to them in a way that allows them to really take advantage of it using their experiences and their goals for the study. And so one way we're approaching this is through developing different levels of data. So some collaborators may only want to take our identified metabolites and work with those and perform their analysis with those. And then others may want to use all 100,000 signals or whatever we're detecting and perform these truly untargeted approaches. And at the same time, we're developing libraries and databases that allow us to support large-scale chemical identification. So I haven't discussed it in detail, but in order to confidently identify a chemical that we detect on the high-resolution mass spectrometer, we often need to buy an authentic standard, which is just that pure compound, and run it on the instrument and see if it matches what we're detecting in our samples. And so we want to really support large-scale chemical identification and make it possible to provide a large number of confident or identifications from our data. And so to do this, we're building these large chemical libraries. To date, we've analyzed about 1,200 standards across our different LC platforms, but we also have a very large number of different environmental chemicals and other chemical libraries that will be analyzed in the near future. And these include all 4,500 ToxCast phase 3 libraries. We have different microbiome libraries and drug libraries. And when finished, we'll be establishing a high-resolution mass spectrometry database of over 7,000 unique chemicals that represents many different exposures, drugs, and commercial products. And our goal is hoping that this library essentially allows us to support rapid identification of thousands of chemical studies in Exposome, or thousands of chemicals in Exposome studies. And so I just wanted to provide a few examples of how we're applying some of these analytical strategies and how we're using them to better understand different disease outcomes. And so this is an example of a precision exposomic study that I worked on when I was at Emory and continued to work on analyzing these results when I moved to Mount Sinai. And so in this paper, which was a collaboration with Kostas Lazaridis, who is who's at the Mayo Clinic, Dr. Jones, who's really led the pilot, and Dr. Miller before he went to Columbia. And the goal of this study was to understand environmental factors of primary sclerosing cholangitis, or PSC. Now, PSC is a liver disease of unknown etiology. There's some genetic risk factors that may increase risk for developing PSC, but otherwise it's, the disease variance is really unknown. And so through this collaboration, our goal was to essentially use different exposomic approaches to understand different aspects of PSC. So the first was to use these strategies to identify environmental associations associated with the disease. Next, we wanted to see how metabolic pathways changed in response to the disease process. And finally, we wanted to see if we could link those exposures to the changes in those pathways to better understand how exposures could begin or start this transition to the disease state. And so within the exposome-wide association study, we applied the combined GC and LC profiling, and we were able to identify a number of different exposures that were associated with PSC. Some of these included known pesticides, preservatives, and industrial chemicals, but we were also able to use our untargeted data to identify a number of different pesticides and other environmental chemicals that we weren't initially looking for. And an example of one of our identified compounds was this pesticide turbocarb, which is used for a number of different applications and was very, very strongly associated with PSC in this population. We then used the metabolomics or endogenous pathway alterations to understand what are the metabolic alterations underlying the disease itself. We were able to find changes in sterol metabolism, inflammatory pathways, and a number of different liver-related metabolic processes. The changes in bile acid were consistent with what's known about the disease process, but the accumulation of urea cycle intermediates and was a novel finding within this study. And that's important because PSC is really identified or characterized by this progressive liver damage, and accumulation of these toxic urea cycle intermediates could potentially be one mechanism contributing to propagation of the disease. And finally, we integrated these biological alterations with different exposures. And we were able to find that a number of them were linked to changes in metabolic pathways, again, consistent with the disease process. We saw changes in pesticides and microbiome-related pathways, as well as changes in oxidative stress and sterol pathways. And so this was originally designed as a pilot study, but since this original pilot study, it's been extended to a much larger research project that's being led here at Emory by Dr. Jones, where they'll be analyzing PSC cases and controls in thousands of samples to try and, again, better understand what are the environmental drivers of the disease. And I'm going to skip these because I'm running a little low on time. I want to leave time for questions. But I just wanted to quickly highlight some of the cardiometabolic disease studies that are currently ongoing in our lab. So we collaborate with a number of different researchers really all over the world. And through these collaborations, we've become involved in a number of different very, very interesting diseases using these exposomic approaches to study metabolic disease, type 2 diabetes, and other obesity-related outcomes. So some of these include looking at exposome drivers and risk factors of metabolic disease in premenopausal women. So we'll be analyzing about 3,500 samples from the SWAN cohort, and all of these samples were collected prior to diagnosis, so we can see how exposures may predict increased risk of metabolic disease and type 2 diabetes later in life. We have a number of studies looking at exposures and type 2 diabetes risk factors in different ethnic populations. So we have studies in New York looking at African Americans and Hispanics, as well as in USC, in California, where we're focusing specifically on Hispanic populations to understand how, again, exposures may be driving increased risk of diabetes in those populations. We also have a number of studies where we're using a bariatric surgery model to understand how exposure may influence weight loss following bariatric surgery. So a lot of these compounds we're interested in measuring accumulate in adipose tissue. So there's some question of, as these people rapidly lose weight, are they releasing more of these pollutants and potentially changing or decreasing the weight loss that could be possible following the intervention. And I also want to highlight some of the studies we're trying to start up now that are incorporating our more novel exposure measures to study how these may increase risk for, risk factors that increase metabolic disease risk later in life. There's a lot of risks in that sentence, but and so this is just an example of the DIMPLE cohort that we're trying to develop with Tanya Alderetti, who's based out of the University of Colorado at Boulder. And this is a very, very interesting study because it will be the first birth cohort that was specifically designed to try and understand how microplastic exposures may influence the gut microbiome development and growth trajectories in infants. So there's a lot of evidence suggesting that increased growth early in life leads to increased risk for obesity and metabolic disease later in life. So we're trying to study, exposures that could occur during this time period and increase those risk factors. And so in this study, we're planning to enroll recently born infants and their mothers. We'll be measuring microplastics in breast milk and stool and different birth tissues, and looking at how they alter the infant gut microbiome and systemic inflammation in the infant. And finally, one of the most important aspects of this study, I think, is that we're doing a survey to identify what are the lifestyle factors, and social determinants of health that predict your microplastic exposures. So I'm sure, all of you have heard about microplastics. They're in the news a lot. You read stories about how you eat a whole credit card every year or something. But the challenge we face is there's just so little known about them and so little known about what exposures we're experiencing from them. And so the hope of this study is really to identify what are the key variables driving microplastic exposures in these infants and how may that affect their health. So just in closing, I'll spend about 2 minutes talking about this idea of the Human Exposome Atlas project. And so as you saw from the presentation, these metabolomic and exposomic measurements essentially provide us a measure of the continuum from an initial exposure to the disease outcome. So we have the ability to characterize both parent compounds and biomarkers of many of exposures using these untargeted approaches. And then at the same time, we can also characterize the exposure and metabolic effects of many, many different diseases. And so we really should be thinking about how we can integrate this information and create a cumulative relationship that allows us to look both within and across diseases. And as an example of how we're doing this, we're moving forward with this in cancer right now and really trying to develop a cancer exposome atlas. And through, to develop this, we're essentially analyzing, over 15,000 samples from many different cancers. We have thousands of samples from lung cancer. We have, thousands from lymphomas. We're measuring about 8,000 breast cancer samples in total. And these are all samples that were collected prior to the individual being diagnosed, which is very, very challenging to get and can be very, very rare. And the reason we want these pre-diagnostic samples is that as somebody becomes sick, their lifestyle changes, their diet changes, their exposures change. And so oftentimes we're concerned with reverse confounding when we're working with cross-sectional studies. But by looking at these pre-diagnostic samples, we're essentially measuring their exposome years before they became sick. And so we can relate that to future disease risk and also look both within and across these different cancers. And so there's a few different challenges we face in this, but really, I hope I've convinced you, or I'll stay here and draw it on the board and convince you further that we've made a lot of progress in working towards this idea of developing a human exposome atlas. So we've developed, the technology has advanced in, huge amounts since this first started about, 10, 15 years ago. We have tremendous sensitivity on our instrumentation, tremendous resolution. We've made a lot of advances in compound identification using both computational approaches and experimental approaches. We've seen, tremendous improvements in our throughput and capacity. Our current assay in the lab will allow us to analyze about 20,000 samples per year using one set of instrument pairs. So if we scale that up, we can increase those numbers even further. We have the study populations available. So we have things like the Million Veterans Project and All of Us, which will really allow us, if we can perform exposome profiling, to do a very comprehensive analysis of many, many different populations. We have chemical coverage. So we've developed different strategies and complementary analysis methods that allow us to measure many different chemicals. And finally, we have, made, although we're not all the way there, a lot of developments in data science and assembly of these resources. And so these are really things that are allowing us to move forward. But there are a number of challenges that we still have to overcome, but I like to think of these more as cautions rather than stop signs or red lights. And that's why I have the caution light up here, because there are things that we need to consider and address, but I don't think they should stop us from moving forward. We shouldn't get so caught up on what we can't do rather than what we can do that, we shouldn't let these things stop us, and it should allow us to move forward. So one of the challenges we do face is we don't have a consistent ontology for what we're measuring in our exposome studies. Some labs use, one chemical database, we use another. So what is that consistent ontology that we can establish to know that if I measure something here and Dean measures something in his lab, they're the same thing? We don't have criteria for harmonization. So how do we know if the data I'm generating is good enough to be harmonized with Dean's data or vice versa? And so we have to have essentially thresholds that we say this data is suitable to be harmonized into the atlas. We don't necessarily have standardization. So a lot of labs develop these exposome assays depending on what their primary research focus is. But I think it's important that we identify what are the standard strategies that we need to use to really move these forward and know that we're getting consistent data across labs. This is more of a question that we all have to think, kind of look deeply in ourselves. Do we have the motivation to do this? Is a human exposome project something that will benefit society? Will it benefit healthcare? Is it worth putting resources into this? I think it is, but all of you may not. It's really, is there the motivation to invest and move this forward? Do we have clear metrics for success? So the Human Genome Project was first assembled, or the first science paper came out in 2001 or 2002, I believe. But Nature just published a paper last year saying the Human Genome Project is finally complete. So what do we consider a success? When can we say the human exposome is complete, or will it ever be complete? And finally, do we have the resources? And I usually put this in for whenever we're talking to NIH program managers and officers, but will we have the funding resources available to us to invest in the technology to develop this further? When the Human Genome Project started, the ability to complete the human genome was not there, but the technology rose to meet the need because of the investment and the resources that were put into it. And that's, will that be available for us in exposome research? So in summary, there's this need to include improved measures of exposure and effect for determining the role of environment in disease. The combination of gas chromatography and liquid chromatography, high-resolution mass spectrometry, really provides us with a unified platform for exposome-wide association studies of disease. Through improvements in these methods and automation and the strategies we're using, it's now really possible to do large-scale exposome epidemiology. We shouldn't be thinking of studies that are 200, 300 samples, but thousands, if not tens of thousands of samples. And so, development of these large-scale platforms now make possible development of these exposome disease atlases. And I just wanted to highlight that, our laboratory is right over there in CNR. Our instruments are up and running. Thanks to Maddie and Catherine, we are, running assays. So happy to discuss collaborations or talk about developing projects. And a lot of this work that was shown here was done while I was at Mount Sinai. So I'm really excited to be bringing it here. A lot of the automation and large-scale exposome work was developed by Brooklyn, Brismar, and Catherine. And so, they really advanced our ability to do this type of large-scale profiling. Of course, I'd like to thank funding, and again, really happy to talk about collaborations and answer any questions in the 3 minutes we have left. Sorry if I went a little late, but.
Thank you, Doug. It's kind of a whirlwind tour of all the great work you do. I can think of numerous projects. I'll get back to you on it. I want to talk to you about a diabetes exposome atlas in particular. We'll talk about that. But start off with one very practical question. What kind of samples do you need to do this, and what's the cost of doing it? And can you use dried blood spots to do these assays?
Sure. So, most of our assays are based upon plasma or whole blood. The LC assay requires, I think, 40 microliters, the GC 100. So typically we say we'll use 150 microliters, but request 250 microliters or more. The cost right now is $205 for one of the assays, or $375 for both, because there's some economy of scale when we're aliquoting things together. And yeah, we absolutely can analyze dried blood spot samples. We've done it previously. Dean's analyzed a lot of dried blood spots. We haven't set the protocols up in the lab here, but it's something we're planning to do and can be easily done. We have a number of projects that are using dried blood spots, so yeah, they're a very valuable sample when it's not possible to collect, when it's not feasible to collect whole blood or plasma samples.
Thanks. Other questions for Doug? There's about 2 minutes left. Arshad?
Doug, it's Arshad. Can you hear me?
Yep.
Yeah. So thank you. That was a great talk, a wonderful overview. Had many questions, but just start with a couple of them. You talked about getting from the metabolomics readout details of endogenous processes, so-called, and then microbiome related pathways, and then exposome, meaning environmental toxins and things. So is that sort of a flow that is now well established? I'm talking about Dean's lab when I work with, who I work with. And then the second question was harmonization that you also addressed, different labs doing things a little differently, different machines. And how would you set about that harmonization? And this is relevant because one of our projects is being done, it's a CAR study where samples are being done in India, lab there, using another high throughput technological platform, but not exactly Orbitrap. So how would you go about doing that? Sort of having a few samples run in both labs and seeing what's common, that kind of thing.
Yeah. So to address your first question, Dean's actually right here, so I'm sure he can chime in if I answer this wrong or whatever. In terms of identifying the exposures, identifying the metabolic alterations, identifying microbiome related pathways, we're somewhat limited in those by our databases. So we're using databases to really identify what could be coming from the microbiome, what could be endogenous, what could be an environmental chemical. We've tried to make it so that we have our individual libraries. So like I said, we have a microbiome chemical library, we have our exogenous chemical libraries, and that allows us to kind of separate those different classes, but it can still be very challenging to apportion a metabolite to a specific process. So you may have metabolites that could be a microbiome metabolite or could be an endogenous metabolite. And in that case, we can't necessarily say whether they're from the host or from the bugs themselves. And I think that's something that we're still will need to continue to develop and thinking through. And in fact, we have a project that Catherine, a member of my lab, is working on, where we're essentially collaborating with other investigators who are taking different microbiome suspensions. So they're enriching for different bugs, adding different chemicals to them and seeing what metabolites are being produced by those. But it's still, we have ways of parsing out these different aspects of the metabolome, but there still is some uncertainty in it in terms of the workflow and how to best do that. In terms of harmonization, what's the saying? It's, an ounce of prevention is worth a pound of whatever. When it comes to harmonization, it's a lot better to be thinking about harmonization from the beginning rather than trying to lay harmonization on top of something that's been done. So one thing that we've tried to do in my lab and for some of the big consortiums we work in is develop a standardized pooled sample. We have two different pooled samples we run, one of young individuals, one of old. And we made a lot of it so that if we're trying to harmonize with another laboratory, we can send them that pooled material. They can analyze it in their batches, and then that gives us a common reference place. Dean and I have been having a lot of discussions, and Dean's been really spending a lot of time thinking about this harmonization challenge. There are a lot of difficulties in doing that. And the most simplest, one of the probably easiest to address, but one of the biggest challenges right now is just that ontology that I mentioned. So, I may call something acetyl L-cysteine, and somebody may call it acetyl N-cysteine. And, currently, if we take those and try and align them computationally, they're seen as two different compounds. And so, finding ways to think through that ontology challenge is also very difficult and something that I think we need to come to a consensus on in the research community. I don't know if you have anything to add to that. Yeah.
when you thought about that and even dabbled a little bit in creating a prototype, but it seems as though that's going to be necessary to drive it forward.
Yeah, absolutely. And so that's kind of, we have started working on it with some of our obesity studies, right? So we're trying to integrate the data from our bariatric surgery study to the SOLAR study, which is youths from California, because we're interested in seeing how PFAS may influence metabolic changes that could lead to risk factors for these diseases. And to be honest, it's been challenging at this point. So I'm an engineer and half an analytical chemist. I'm certainly not a data scientist. And so thinking of how we can best assemble it and prototype that is very difficult, even to the point of what are the most appropriate database structures or languages that we can use to essentially tie all that together. So in terms of building a prototype, we don't really have one yet, but I think within the next year, we'll be analyzing 3,000 breast cancer cases from the multi-ethnic cohort in California, another 4,000 from the sister study, another 3,000 lymphoma samples, all analyzed on the same platform using the same approaches. And I think we just have to start from the lowest common denominator, the easiest aspect of that prototype. So can we create this atlas and make it a resource for the studies that we're running, and then start thinking about how we can add different assays or different platforms onto that as well. Yep.
Thank you for that presentation. As a ardent student in biochemistry and nutrition field, my question is, you talked about the different databases. I think a lot of it's being driven from what I would call metabolism, organic chemistry, organic compounds. And then you have sort of, sorry, I'm too old here, but then you have your inorganic. And I'm just curious, do you see gaps in those databases? Because it's when these metals, for example, get incorporated into organic systems. Just wanted to get your thoughts on that.
Well, so that's something, and even the question we had about the microbiome and differentiating endogenous metabolites from the microbiome, the whole concept of metabolic pathways itself is somewhat artificial, right? Because if you look at metabolism, it's a complex interaction among all of these different metabolites. And then somebody said, this is tyrosine metabolism, this is phenylalanine metabolism, but there's inputs and outputs that kind of span the pathways. And so, thinking of it in terms of these more historical databases is very challenging. Now, in terms of the metals, that is something that I have not done a lot of work in. So I primarily have focused on, measuring these organic molecules. We do have some studies where we're looking at how folic acid intervention influences arsenic exposure. So how do dietary alterations in Bangladesh affect total arsenic exposure, which is kind of getting to that. But I think in terms of metal metabolome relationships, that's really something that they've been working on for years in Dean's lab, and I'll have to kind of,
Metal is an organic matrix. So I just was curious. I see that as the intersection of well.
Unfortunately, this is a subject that's going to create holes in the wall that people are going to bang their head off the wall, because it is that difficult. It's among the more challenging things, and as Doug indicates, we've worked on it for years, and I have to say we haven't made a whole heck of a lot of progress on that.
And even from the air pollution, we always see arginine associated with air pollution exposures, and the arginase enzymes are all manganese-containing enzymes, and so there's a lot of interesting interactions.
Yeah, that's why I was like,
It's the potential. There's the potential there, but we don't. We just haven't been able to sort it out. I mean,
The question is related to gene-environment interaction. So yes, participants are saying that about 15% of a disease can be attributed to genes and gene-environment interaction on average. Is there like variability in terms of different tissues getting different levels of deposition because of gene-environment interaction? And the reason for asking that was just to understand how population stratification in genetics might influence whether a finding is transported from one population.
Yeah, so in terms of looking at deposition or tissue effects, I mean, that's something that we are still trying to explore further. I mean, it's very, very likely that certain tissues are kind of differentially affected by exposures, and of course how you detoxify those or other genetic variations could kind of influence how they're affecting that tissue. One of the ways that we've kind of approached this is through some of our women's health studies, where we're looking at different samples that tell us more about like reproductive effects of many of these exposures. But there is the potential for differential exposure in tissue to affect those. But the challenge we always face is, what's the best proxy we can get as a measurement for understanding exposure? And really, blood in these biological tissues tend to, on average, give us somewhat of a good prediction of these. In terms of population stratification, so if I understand correctly, are you saying that there could be, different genetic subpopulations that are affected by an exposure differently, or you have, different exposures within a population? I'm sorry if I misunderstood that. Yeah.
So, what we might be finding, if there's a substantial gain interaction, could be the effect of the gene over the effect of exposure.
Yeah.
Especially if you measure the exposure in an ethnically similar population.
Yeah.
Without broad migration.
Yeah. Yeah.
So it might be hard to partition like that.
Yeah, absolutely. And I think that's why, that's what drives a lot of our need to develop these large scale approaches. So, I think we need to get to a place where we can analyze 100,000 samples from 100,000 individuals and, and then have paired genomics data. And so look at what are the gene by exposome or gene by metabolome interactions that may be driving a disease. But we can't get there with the current studies we have. We need large studies to be able to do that. I saw another question.
Yes, I'm thinking a little bit in a different direction. I was interested when we talked about motivation, measure holding this tension, a lot of different pathways to do your work, which is impressive, and I appreciate it. But I wanted to ask you about resources and competition for collaborations. Some people just really know that I love neurons to be at the forefront of whatever we do. So I'm curious about where are the other players in this field and those collaborations that you referred to, because it seems like it's been taking a lot of significant collaboration to put together this kind of massive compilation of data. And then I'm curious about what do you need, what are the resources for blocks to your work?
Sure. Yeah. So, and actually, that's a very conveniently timed question, because NIEHS is releasing an RFA about a global exposome coordination center. So how can you tie efforts in the US to what's going on in Europe and find, common ground on how to best develop exposomic methods? In terms of, where's the main exposome research happening now? I mean, Rollins really is at the forefront of exposomics. They were the first US exposome research center, and when Hercules was funded, and have continued to really be at the forefront of exposome research, thanks to Carmen's efforts, Dean's efforts, everybody who's really been working in that field.
Noticed that my office was next door to Gary's through all that period of time. So I lived up close and personal, waiting for the grants to come through, actually for me, waiting to be able to show this to our friends and potential donors. So I'm particularly interested in that. So that's great. There's something coming down the line. There may be private sources too.
Excellent.
But we certainly need to know about that. This is probably an offline conversation, but I've always been fascinated in Hercules and Exposome and what we've learned about that. I'm glad to know we're still, I knew Dana and Gary would lay it out front, but it takes a lot to keep up too.
It's really still for the health, I think. Honestly, and I think that is the opportunity. I mean, Doug already has his fingers in so many pies, but there are a lot of resources. The issue is, as I was suggesting, the prototype, it really needs engagement of many more individuals to help, because the tasks are major.
Well, which I'm sure means that you really do want to build this collaboration. You know where to place them to it all.
Yeah.
So I'd love to hear more about it, maybe not what this minute,
Yeah.
more about how you're building this networks and how they work and
Of
what
course.
partnerships are, and what do we need to do here to keep us.
Great.
close to the-
Yeah, and I actually came from a model. Mount Sinai has an Institute for Exposomics Research, and that was almost entirely funded through fundraising efforts in New York. And so, we had a lot of expertise kind of working in that regard, but that was also a really nice opportunity because they hosted a lot of public events and really tried to bring, more simplified, kind of better understanding of what the exposome is and all the exciting research we're doing. But yeah, it's there are definitely some main sites like Sinai, Columbia, where Gary is now. But one of the things that's really been an advantage for me, and Gary is involved in this as well, is that we're tied into the European Exposome network. So we were the only 2 US partners that were funded as part of their big European Exposome network. And my microplastics project, which I didn't really talk about, was the only US partner funded by the EU Horizon 2020 to contribute to that. So it's what's been really nice is that we're establishing all these global connections. I'm going to South Africa next month to talk at this big prostate cancer meeting because they found that they've been studying genetics of prostate cancer in African men, and the genetics is not explaining the increasing rates in early onset disease. And so they want to talk about the exposome and how that could be incorporated into their study designs. And so, yeah, there's really, I think right now is a critical moment for us to continue pushing it forward. And, I'm really fortunate or feel very fortunate to be in a place that's been so supportive of, staying at the forefront.